topic that I'm going to discuss in this video is about the ultrastructure of bacterial cell. The structure of bacteria is known for its simple body design. Bacteria are single cell microorganisms with the absence of the nucleus and other cell organelles. Hence, they are classified as prokaryotic organisms. This slide shows the overall ultrastructure of bacterial cell. The structures present outside the cell wall includes flagella, capsule, pili, and sheath. And the structures present inside the cell wall includes cytoplasmic membrane, nucleoid, mesosome, ribosome, and plasmids. Appendages. Appendages are the structure present outside the cell. First, we are going to discuss about flagella. Flagella are present in some rods and spiral forms, and their function is motility. And the origin of a flagella is the cell membrane of the bacteria. It is attached to the cell membrane by hook and basal body, which consist of set of rings which differ from gram-positive and gram-negative bacteria. The gram-negative bacteria consist of two sets of ring and rods, that is LPSM rings and rods. Example E. coli. Whereas in gram-positive bacteria, two rings are present, S and M, and rods are also present. Example Bacillus megatherium. Next type of appendages we are going to discuss here is about fimbriae and pili. Fimbriae are shorter than flagella and striated, smaller hair-like appendages present only in some gram-negative bacteria, and their main function is to adhere, and it does not involve in motility. It is one of the invasive mechanisms present in bacteria to cause diseases. The next one is pili. Pili is the sex factor present in bacteria. It is necessary for bacterial conjugation, resulting in the transfer of DNA from one cell to the other. Next structure that we are going to see is bacterial cell wall. Bacterial cell wall is made up of mucopolysaccharide, that is peptidoglycan. This peptidoglycan is made up of N-acetylglucosamine and N-acetylmuramic acid, and it also contains a tetrapeptide, namely L-alanine, isoglutamine, lysine, and alanine. The entire cell wall structure is cross-linked by covalent bonds, which provides the rigidity necessary to maintain the integrity of the cell. N-acetylmuramic acid present in bacterial cell wall is unique to prokaryotic cells. This picture shows the cell wall difference between gram-positive and gram-negative bacteria. Gram-positive bacteria contains a thick peptidoglycan layer, and inside of which is a cytoplasmic membrane. Whereas in gram-negative bacteria, peptidoglycan is present in the middle, outside of which is lipopolysaccharide and proteins, and it also contains a periplasmic space. Next structure we are going to see is cell membrane. The function of cell membrane is to control the permeability of movement of materials, and it transports the protons required for cellular metabolism, and it also contains the enzymes to synthesis and transport cell wall substances, and it also secretes hydrolytic enzymes and regulates the cell division. Bacterial cell membrane is similar to eukaryotic cell membrane. It contains a phospholipid bilayer and protein. Bacterial internal structures. Cell cytoplasm. Cell cytoplasm is a dense gelatinous solution of sugars, amino acids, and salts. And 70 to 80% of cytoplasm is made up of water, which serves as a solvent for materials used in all cell functions. Next internal structure present in bacteria is chromosome. Chromosome is a single circular double-stranded DNA molecule that contains all the genetic information required by the cell. DNA is a tightly coiled structure surrounded by protein, which is called as nucleoid. Next structure that we are going to see is plasmids. Plasmids are small circular double-stranded DNA. They can be either free or integrated into the chromosome. It gets duplicated and passed on to the offsprings. Plasmids are not essential for bacterial growth and metabolism, but it is an extra chromosomal material present in bacteria, and it may encode for antibiotic resistance, tolerance, enzymes, and other toxins. This plasmid is very important in genetic engineering for manipulating the DNA from one cell to the other. Next structure present in bacteria is ribosomes. Ribosomes are made up of 60% of ribosomal RNA and 40% of protein. It consists of two subunits, large and small. Prokaryotic ribosomes differ from eukaryotic ribosomes in size and number of proteins. Site of protein synthesis present in all cells. This picture shows the ribosome 70S, which consists of larger subunit, which is 50S, and small subunit 30S. Next internal structure of bacterial cell is mesosomes. Mesosomes are large invaginations of the plasma membrane and irregular in shape. It increases the membrane surface, which may be useful as a site for enzyme activity during respiration and transport. And it participates in cell replication by serving as a place of attachment for the bacterial chromosome. The next structure present in bacteria are inclusions. Inclusions are not separate by a membrane, but it will be distinctly present in bacteria. Next structure present in bacteria is chromatophore. Chromatophores are present only in photosynthetic bacteria and blue-green algae, whereas generally in all prokaryotes, chromatophores are absent. Endospores are the special structures found only in bacteria in both gram-positive and gram-negative. The spore-forming bacteria's example includes Sporobacterbacilli and Sporosarcinae. They have medical importance. Most of the bacteria have medical importance, and the position of endospores is median, subterminal, or terminal lanes. And their main role in bacteria is to provide resistance to heat, UV, and other chemicals. So, thanks for watching the video.